Big thank you to Nebula for sponsoring this video. It is August 8th, 2001. President George W. Bush is about to give his first ever nationally televised address. The topic may surprise you. It's still just 8 months into his first term. So far, the biggest issues of his presidency have been corporate tax cuts and education reform, neither of which warranted an intimate sit-down with Bush at his ranch in Texas. The tragedy that would go on to define much of his presidency was still an entire month away. He had something else on his mind.
Good evening. I appreciate you giving me a few minutes of your time tonight so I can discuss with you a complex and difficult issue, an issue that is one of the most profound of our time. The issue of research involving stem cells derived from human embryos is increasingly the subject of a national debate and dinner table discussions. My administration must decide whether to allow federal funds, your tax dollars, to be used for scientific research on stem cells derived from human embryos. I strongly oppose human cloning, as do most Americans. We recoil at the idea of growing human beings for spare body parts or creating life for our convenience. And while we must devote enormous energy to conquering disease, it is equally important that we pay attention to the moral concerns raised by the new frontier of human embryo stem cell research. Even the most noble ends do not justify any means.
And just like that, the White House issued an executive order to ban federal funding for new embryonic stem cells. It was a decision that would almost immediately kill all US momentum in the field. And in Congress, a battle was raging on whether they should ban the practice entirely. If not for the events of September 2001, the stem cell issue may have gone on to become one of the defining political controversies of the Bush Jr. years. And it was all kickstarted because of a 5-year-old with 3 moms. Dolly was born on the 5th of July 1996 at the Roslin Institute in Scotland, although she wasn't announced to the world until February of 1997, at which point she was about 7 months old. Dolly is also a Finn Dorset sheep, and her birth resulted in more laws being passed than perhaps anyone else in history. You see, Dolly is a clone, an exact genetic copy. Your body is made up of cells. Trillions of blobs that make up your muscles, your bones, your organs. Inside those little blobs are a bunch of chemical gizmos, let's say. The most important gizmo is the nucleus, the brain of the cell. Inside the nucleus is a full copy of your genetics, your DNA. It's an acronym that definitely stands for something, but you don't really need to know what. DNA is essentially a gigantic instruction list in the shape of a double helix. And in that double helix are a bunch of molecular pairs, A and T, or C and G. It's one or the other. We call this a base pair. Humans have approximately 3 billion base pairs, and the precise order of all those A's, T's, C's, and G's determines stuff like your hair color, eye color, how tall you are, whether cilantro tastes like soap, over 20,000 different genes. Now, when 2 animals reproduce, most of the time it goes like this. 50% of the DNA is contributed from a sperm cell and 50% from an egg cell. The DNA combines and you get a new full set of DNA that's a combo of both parents. That single cell then multiplies, going from 2 to 4, 8, until it can potentially become a full organism. Now the crazy part is that 99.9% of your DNA is shared with every other human. And you and your parents, the closest genetic match most people will ever have, share 50% of that very last 0.1%. That last decimal point is what separates you from everyone else. The exception to that would be a clone. Like I mentioned earlier, Dolly had 3 moms. One mom provided the egg cell, with its DNA removed via a small needle, leaving behind an empty container. The second mom provided a nucleus, with a full 100% copy of its DNA. That nucleus was inserted into the empty egg cell, which was then stimulated with electricity to artificially kickstart cell duplication. Then, Pretty much the same as before, that cell multiplied, 2, 4, 8, until it was stable enough to become implanted into the womb of the third mom, a surrogate. The third mom was pregnant for the usual 5 months, and then she gave birth to Dolly, who was a perfect genetic match for the second mom, the one who contributed the DNA. Dolly was one of those rare scientific breakthroughs that became a global news sensation, a living, breathing clone that you could visit and pet, a marvel of human ingenuity, like something out of science fiction. Except Dolly was not the first ever clone. In fact, she's not even the first cloned sheep. Humans have been cloning plants for literally thousands of years, and when it comes to animals, the first ever cloned tadpoles were made in the 1950s. So why was Dolly so special? Why now? It's because Dolly was the first ever clone from an adult cell, not an embryo, and that had huge ramifications for medicine, human medicine. The cells in your body have specific functions. Bone cells are rigid and provide structure. Muscle cells are flexible and allow for movement. Nerve cells send electrical signals, connecting your brain and the rest of your body. But Dolly's existence proved that any adult cell has all the necessary genetic information stored in their little nucleus to revert back to an embryonic state. A state where all possible cells are part of a vast, branching tree of potential. And at the bottom of that tree, the stem, is the embryonic stem cell. Stem cells are a uniquely potent cell that has the remarkable ability to repair our bodies. Think of the Pokemon Eevee, a generic base template that can be molded into any type with the right environment and stimulus. With stem cells, you can create bone marrow cells to treat leukemia. With stem cells, you could create pancreatic cells to produce insulin for diabetics. With stem cells, maybe you could even grow someone a new kidney. Medicine at the turn of the 21st century felt like it had endless potential, all because of a sheep named after a country music star. The looming question, though, was how do we do this responsibly? There are 2 types of cloning: reproductive cloning and therapeutic cloning. The first is what Dolly is, a way to make copies of full organisms. Therapeutic cloning is smaller in scope, a way to make copies of genes, tissues, maybe even full organs, with the goal of treating or preventing disease. One of these was broadly unpopular, especially when it came to humans. The majority of scientists agreed it was too risky, too morally dubious to try and clone a human child. Nearly every major world leader expressed their desire for a ban on cloning humans, including Bill Clinton. Therapeutic cloning, on the other hand, was much harder to draw a line for. With it, you could create stem cells that had the potential to save millions of lives. But because it required human embryos, that made it a deeply politicized topic, and the Bush White House intervened. And American scientists had strong opinions on this. I think there is every reason to believe that if passed, this kind of prohibition would not be effective. I think that thousands of years of human experience have shown us that governments cannot bottle up human progress, even when you want to. We will be sitting here with the best scientists in the world watching things on television. In his speeches, Bush evoked worst-case scenarios that included organ farms, made-to-order children, and desperate women pressured into selling their eggs. But his critics argued the opposite. The best way to control the research is to fund it by the federal government, because then you create rules. This is true. Even without Bush's ban, the FDA was already regulating cloning techniques. As soon as any researcher so much as touches a human egg cell, there's a paper trail and dozens of eyes on them. By opting for an outright ban instead of regulating it, Bush was ensuring one thing. People are still going to do it. They're just going to go to different countries. And that is where our story takes us. In July 1997, a few months after Dolly was revealed to the world, Thailand devalued its currency. The 1990s had seen several East Asian countries rapidly industrialize and expand their economies with absurd growth rates that seemed far too good to be true. But as 1997 came around, global exports slowed, particularly in the semiconductor market, and foreign investors saw blood in the water. That year, Thailand's stock market completely tanked, and the crash initiated a cascade that brought down the economies of several nearby nations. Malaysia, the Philippines, Singapore, all were hit with recessions. Indonesia even forced its president to resign. But our story truly begins in South Korea, which was finally feeling the shockwaves by October 1997. Although the exact mechanics of the crisis are a bit beyond our scope here, South Korea's economy was by and large dominated by massive corporations known as chaebols. Brands like Hyundai, LG, and Samsung. Behemoths that were seen as too big to fail. At least until they weren't. They'd been taking on aggressive and risky foreign loans, expecting the government to be their safety net. But something had to give. Before the crisis, South Korea had 7 major automobile companies. Only 2 emerged from the crisis unscathed. By 1999, of the top 30 chaebols, 11 of them had gone into bankruptcy or bankruptcy protection. Included in that number was the Daewoo conglomerate, which at the time was the largest corporate bankruptcy in history. By 1998, 1.5 million South Koreans lost their jobs. All in all, it was a terrible way to be ending the 90s for a nation that had suffered so much in the last century. The first half of the 20th century saw Korea brutally colonized by the Japanese, only to then be further occupied by the Americans, leading to a devastating war that divided the country permanently in two. What followed in the South was decades of military dictatorships, political unrest, and now an economic crisis that was undoing the last decade of relative stability. To put it simply, things were bad, and the timing couldn't have been worse for the sitting government. The crisis began to snowball just weeks before the 1997 presidential election, which saw the incumbent party ousted by a slim margin. The incoming president was longtime neoliberal candidate Kim Dae-jung, and a priority of his administration was to figure out how to make South Korea a major player on the global economic stage, so this could never happen again. It didn't help that the country was being crowded out by 2 of its next-door neighbors. To the east, you had Japan, which had a clear edge when it came to award-winning scientific research and consumer electronics. And to the west, you had China, which was undergoing unprecedented population growth and was well on its way to becoming the cheap manufacturing hub of the entire world. During the 90s, the South Korean government had found some success by making significant investments in semiconductors and IT, but that alone had not been enough to shield them from the crisis of 97. No, it needed something else. A cutting-edge technological niche, one that the government could devote millions to, one that South Korea could make part of its national brand for the new millennium. And so the government looked at Dolly and said, we can do that. But it wasn't enough to just throw millions of dollars at biotech research in general. South Korea had already been doing that since the 80s. They were up against Japan, China, as well as the Western powers like the US and the UK. They would never win in a battle of who can spend the most money. They needed something more than that. With their limited funds, they crafted a strategy to support a select number of star scientists to elevate them as symbols of national pride, to inspire hope. Economic growth is one thing. GDP, inflation, unemployment rates, that's all well and good. But statistics and acronyms aren't tangible to the average person. Koreans needed someone to latch onto, whose research would make headlines in the newspapers, whose face they would come to recognize on TV. They needed a celebrity. The idea of a national celebrity scientist is not a concept unique to South Korea. All around the world, we often think of science in terms of iconic individuals, lone geniuses, visionaries. We'll often see a project with hundreds, if not thousands of researchers and boil it down to the lone face in charge, a single person who we project our own national values onto. Within the realm of biotech, this was already happening. Around the turn of the new millennium, the Human Genome Project was nearing its end goal, a decades-long task to write down all 3 billion base pairs found in human DNA. Even though this was a goal that dozens of countries were collaborating on, media coverage of the project often framed it as a competition between 2 Western nations, each of which had their own mascot. As the Daily Mail pointed out at the time, in the British corner, devoted in earnest, is John Sulston, 57, who drives a second-hand car and wears battered leather sandals. Surging out on the US side is Craig Venter, 54, a former surf bum who now has a gold Rolex on his wrist and a 70,000-pound yacht called Sorcerer as just one of his toys. Both men appealed to an archetype of what each country viewed as a model scientist, the bold American entrepreneur versus the reserved British intellectual. So who could be that national archetype for South Korea? What scientist should the government pluck from obscurity to lead them into the new millennium? The man they chose was almost too perfect to believe. Hwang Woo-suk is a man whose life story feels almost manufactured. Just 1 year after the first ever tadpole was cloned, Hwang Woo-suk was born. It's 1953, half a year before the armistice that would put an official end to the Korean War. Hwang's family was poor, and they were from a particularly poor region of Cheongdam province. His father passed away when he was just 5 years old, and he had to help his mom tend to his siblings and their 3 cattle. When there wasn't enough food, he resorted to eating tree bark. Even from such a young age, though, Hwang says he dreamed of becoming a veterinarian. Out of his entire elementary school class, Hwang was the only student to receive a higher education. In high school, he was ranked just 400th out of 480 students. He had to work on a farm to just barely afford his tuition. Eventually, though, he worked his way up from the bottom and got accepted to Seoul National University, SNU, to become a veterinarian. From humble beginnings to the most prestigious university in the entire country. He would graduate with a PhD in 1982, with a specialization in theriogenology, aka the study of animal reproduction. Given what he would later become famous for, it's actually a bit surprising that Hwang's early career was not that of a bioscientist, but again, as a veterinarian. He didn't work out of a pristine lab, but of a typical cattle shed on a farm. He wasn't publishing articles to journals, but instead was working on potential improvements to farming, trying to breed cows to produce more milk and bulls to produce more meat. This all changed in 1993, though, when he made headlines for creating the nation's first cow via IVF, in vitro fertilization. IVF is essentially a normal pregnancy that is assisted with laboratory techniques. An egg cell is fertilized with a sperm cell in a petri dish, and the fertilized egg is monitored as it grows into a viable embryo. IVF is often used to help couples who can't conceive on their own by growing the initial embryo in a lab and then implanting it in a surrogate later. On its own, an amazing achievement, but not yet worthy of global attention. By 1995, his goal had evolved, and he began writing books and articles on the potential for cloning dairy cows. Finally, in 1998, it was reported that he had implanted a cloned embryo into a cow, and in February 1999, that cloned cow was born. And it was a spectacle. Hwang, not afraid to get his hands dirty, brought in a TV crew and literally got his hands dirty and helped the mother give birth to the calf. The calf was named Yongnong-yi, which translates roughly to brilliance. Now, Yongnong-yi was not the first cloned cow, it was the 5th. But it was the first clone ever produced by a Korean, and it made South Korea the 5th country in the world to successfully clone a mammal. Not bad for a country that was largely seen as a bit of an outsider in academic circles at the time. The birth of the cloned cow set Hwang on a path to stardom. Soon, he was translating and co-writing books with scientists more famous than him. Next, he was giving interviews and TV appearances left and right, never turning down the opportunity to get his name out there. Finally, it was him sending out invites to journalists and politicians to come visit his farm and see how his research was progressing. Just 3 months after the birth of Yongnong-yi, Hwang announced that he had cloned a species of cow native to Korea, the Hanwoo, which is known for its high-quality cuts of beef. The cow was named Jinni after a famous 16th century Korean poet. At the request of Hwang, the name was chosen by none other than President Kim Dae-jung himself. With the full backing of the South Korean government, Hwang fully transformed from just a humble veterinarian to a full-on distinguished bioscientist. The Ministry of Science and Technology awarded him around $1.6 million to pursue a method of mass cloning dairy cows and cattle for beef. The goal was to mass produce over 100,000 cows by 2008, but ultimately this project ended up failing and it stopped sometime in the early 2000s. Nonetheless, this did not diminish Hwang's reputation. It's rare that a scientist knocks it out of the park twice in a row. This was followed up by him announcing his intentions to clone a Siberian tiger and cows that would be resistant to mad cow disease. None of these promises came to fruition either, but it didn't really matter. Hwang was becoming a master of PR. For his big press releases, he hired professional photographers. When reporters came looking for quotes, he'd personally give them guided tours of his labs to pet the animals he was working with. His announcements were carefully spaced out to keep the public engaged. Whether they succeeded or not was besides the point. He was the talk of the country, and at some point, he'd have another success. That eventually came in 2002, when his team announced that they had successfully cloned pigs. And they weren't even the first cloned pigs in the world, but a success was still a success. Although President Kim Dae-chung had been a proud supporter of Hwang's work, his administration still prioritized IT during the dot-com boom. But as the dot-com boom turned into a dot-com bubble, Kim's admin came up with a more diverse initiative, the 6 T's: information, bio, nano, environmental, space, and culture. Gonna be honest, not my favorite slogan. There's a questionable number of T's there, but you get the picture. The end of 2002 saw another presidential election, and President Kim handed over the reins to President Roh. Since Roh belonged to the same party as his predecessor, his administration inherited many of the same priorities. And of the 6 T's, the one that had the most promise in the short term was biotech. Early into his first year in office, Hwang invited the president to his lab to show him a dog, whose life he had saved via stem cell transplants. Describing the visit, the president said, "I felt that Hwang's scientific achievement was not a technology, but a sort of magic. I certainly discovered the possibility and hope of a new era in Northeast Asia, and the achievement of over $20,000 per capita GNP. Now, I'm so deeply moved that my body trembles, as if I had received an electric shock." Wow, okay, laying it on a little thick, but sure. The government, as well as the media, were carefully crafting a mythos around Hwang. Hwang fit the archetype of what a model South Korean should strive for. Much emphasis was placed on just how humble his beginnings were, how he pulled himself out of poverty thanks to hard work and determination, the classic bootstrap narrative. And even though he was now wildly successful, he never strayed far from his humble roots. Quote, "He rented a small apartment for the last 18 years. He has kept a routine of waking up at 4:30 a.m., going to the public baths, and then meditating for an hour in Goksundo, before going to work at 6:30 a.m. He stays in cheap motels at international conferences, and also flies economy class. His philosophy is that abundance brings laziness. He was so busy, he never even had time to watch TV or read the newspaper. He was often the first in the office and the last to leave. Almost every aspect of his life seemed to be just about furthering the concept of cloning, even his religious beliefs. He once said, 'I am a Buddhist, and I have no philosophical problem with cloning. As you know, the basis of Buddhism is that life is recycled through reincarnation. In some ways, I think therapeutic cloning restarts the cycle of life.' This is a striking contrast to the conversation in America, where religion was one of the fundamental drivers of Bush's funding ban. More than anything, though, Hwang refused to accept personal credit for his biotech breakthroughs. He was always highlighting the spirit of sacrifice of his colleagues, so much so that he often talked about his lab as if it was a family. A personal army of 125 researchers who he described as fighting the war for humanity and curing disease. Their personal motto? Moving the heart of the sky. And the country loved him for it. Here are some quotes from his supporters. "We were suffering a Japanese government for 46 years. So our country, which was independent since 1945, we only have a very short period to develop our country. If a Korean becomes top of the world in their own field, people are very proud. He was going to change our country's image and make South Korea #1 in the world in this sector. We don't have someone to represent us to the world. South Africa, for example, has Mandela." That was the type of figure that Koreans saw in Hwang. But still, he had yet to break out onto the world stage, and there was a simple reason why. At no point had Hwang proven that his clones were actually clones. Yes, you heard that right. His cows and pigs were never genetically tested. He had never once had a paper be peer-reviewed. He held press conferences, photo ops, lab tours, you could even pet the animals themselves, but nowhere did Hwang ever release his data to be analyzed. The international scientific community wasn't ready to shower Hwang with accolades until he could show he was the real deal, publish his results in a respected, internationally recognized journal. So, in late 2003, Hwang finally took a swing. He aimed for the moon. He submitted a paper to the journal Science, one of, if not the most prestigious journal in the entire world, with an acceptance rate of only 7%. And against all odds, his very first paper was accepted. He was about to become the king of cloning. Hwang's groundbreaking paper was titled Evidence of a Pluripotent Human Embryonic Stem Cell Line Derived from a Cloned Blastocyst, which is a whole lot of words, but you really only need 2 of them to know that it was a big deal. Human and cloned. Cloning mammals was considered an earth-shattering achievement when Dolly was announced, and in the 7 years since, there had been several other victories in mammals: pigs, cows, mice, but critically, there had never been a success with primates. So the idea that some unknown Korean team had skipped that step, that he had gone and cloned a human embryo, that was unthinkable. He was years ahead of the rest of this field. Now, of course, he wasn't trying to grow a full human clone. No respectable scientist was. Rather, he was trying to clone embryos to produce stem cells. A new type of stem cell that was the holy grail of medicine. Now, to be clear, your body right now contains stem cells. Unspecialized cells that can self-renew indefinitely, which your body uses to maintain and repair itself. But they're also limited in what they can do. A common example is found in your bone marrow. These are called hematopoietic stem cells. This type of stem cell can specialize into one of 3 types of blood cells: red blood cells, white blood cells, and platelets, each of which carries out a unique function in the body. People who suffer from leukemia often can't produce these bone marrow stem cells on their own, and can be treated with a stem cell transplant from someone else. The limitation here is that bone marrow stem cells can only specialize into blood cells. They can't be used to treat anywhere else in the body. We call these multipotent, a stem cell that can specialize into multiple types of cell, but not all. Even more restrictive types exist, which are called bipotent or even unipotent. They exist in parts of the tree with fewer and fewer branches. Because your cells can never go backwards down the tree, the most useful type of stem cell will be one found near the absolute base of the tree. This would be called pluripotent, opening up nearly unlimited options to help save someone's life. And this embryonic stem cell, as we call it, does exist, but only in the earliest stages of development. In embryos, there aren't yet organs, or blood, or bones, so cells haven't specialized yet. They have the capability to move up the branches of the tree with the help of chemical inducers, slowly turning off unnecessary genes. The first embryonic stem cell line was isolated in 1998, but the applications were limited. If you receive a stem cell transplant, chances are the stem cells are coming from someone else. Their DNA and your DNA are not a perfect match. Your immune system will detect this, and it may reject the transplant. To get around this, stem cell patients often need to take immunosuppressive drugs so that their body won't reject the treatment. Immunosuppressive drugs often leave you vulnerable to a whole host of negative side effects, and you must continue taking them for the rest of your life. But if you could be your own stem cell donor by having your stem cells cloned, that would eliminate the need for the immunosuppressive drugs. It would both reduce the health risks of existing transplants and open up the possibility of things like full organs being grown from your cells. That's what Hwang and his team had achieved. Cloned, embryonic stem cells. The Holy Grail. But the question remained, how had Hwang and his team achieved what no other lab could? The short answer was brute force. Hundreds of failed attempts. The cloning method they had used was notorious for its low success rate. SCNT, somatic cell nuclear transfer. A somatic cell is any adult cell in the body, not sperm, not an egg. Nuclear transfer is the process of delicately taking an adult cell's nucleus and inserting it, again, very delicately, into an empty egg cell. Then the cell is hit with a chemical kickstarter and the cell replicates itself over and over. If you let it keep doing this, you could potentially grow a full clone. But for stem cells, you just need a handful of these cells. You can keep them in a petri dish, take as many as you need for a treatment, and the rest will self-replicate endlessly. This, by the way, was almost exactly the same technique used to create Dolly, which was no walk in the park either. It took them months to fertilize 277 egg cells. Only one survived and grew into an adult sheep. This was the biggest ethical hurdle with the SCNT method, the staggering number of eggs it required. Acquiring eggs from a sheep is significantly easier than acquiring eggs from a human, both from a safety and legal standpoint. Normally just one egg is released from the ovaries in a single cycle, but egg donors are given a hormone injection so that they superovulate, releasing many more eggs than normal, usually upwards of 10 for a single donor. They then undergo an invasive surgical procedure to remove those eggs, which, like any surgery, can have potential complications, some small, some more severe. The key here is that in many countries, egg donors must be voluntary, i.e. unpaid, and they must be fully informed of all of the potential risks. For good reason, acquiring human egg cells is a slow and meticulous process with lots and lots of paperwork. So couple this with the extremely low success rate of SCNT, it's no wonder that human embryo cloning was viewed as a pipe dream by many in the field. And yet this South Korean team had done it. Hwang's paper noted that it had used 16 donors who contributed 242 egg cells. That's quite a high number of eggs, which on its own was impressive. But managing to get a successful cloned stem cell out of all of that was a success rate of 0.4%. Still very, very low, but human cloning had just been upgraded from impossible to extremely difficult. Hwang had fully evolved from a humble veterinarian to a celebrity scientist in Korea, and finally, a leading global researcher in stem cells. Time magazine named him in its list of people who mattered in 2004. At the height of his popularity, Hwang was featured in at least 16 books, where he was the role model of what every young scientist should grow up to be. Hwang had appeared on TV more than any other Korean scientist in history. He was receiving millions of dollars a year in research funding from the government, and Korean Air gave him first-class flight privileges for the next decade. Hwang continued his famous lab tours, but he also started visiting patients in hospitals who suffered from incurable diseases, promising them that his work could potentially save them within the next 10 years. His work had even inspired a postage stamp, following a widely televised appearance where he made the following promise.
I hope singer Karl could stand up again from his wheelchair to show his amazing dance moves soon in this show.
But all of this still wasn't enough. When the average person thought about cloning, they thought about a Scottish sheep. It was Hwang's turn now. When someone thought about cloning, he wanted you to think Korea. The idea of a Korean academic earning the respect of the West is a powerful one. Near the turn of the 21st century, the global gatekeepers in science media were the US and the UK. The two most prestigious journals in the world were the UK's Nature and the US's Science. They were the arbiters of scientific legitimacy. If a Western scientist got a paper in either of those two, it was considered a career milestone. But an obscure Korean like Hwang, that had Westerners raising their eyebrows. Around the time of Hwang's 2004 paper, cloning was undergoing a bit of a rough patch. Dolly the sheep had passed away the previous year at just 6 years old, half the expected lifespan of a Finn Dorset. Although today her passing is thought to be due to a respiratory illness she got from living in captivity her entire life, at the time the prevailing assumption was that clones simply didn't live as long. This not only generated fear among the public about animal clones, but carried over into the public debate on stem cells. Besides Dolly's passing, there had been a few mini scandals that hurt the public image of cloning. Just 1 year after Dolly's unveiling, a completely separate South Korean group used the same techniques to clone a human embryo, but had voluntarily cut the experiment off early at just 4 cells. The team said they had no intention of implanting the embryo into a surrogate, and they also refused to release any evidence of their achievement, casting significant doubt on whether they were telling the truth. And then there are the infamous cowboy cloners, mavericks who ignored the scientific consensus and announced their intentions to create full human clones. An Italian fertility expert claimed in 2003 that he had impregnated multiple women with cloned embryos, followed by a Cypriot doctor making a similar claim the following year. Again, no evidence was ever provided by either man, and they were soundly dismissed as attention-hungry quacks. The spiciest of all, though, were the Raelians. The Raelians are a Canadian-based UFO cult that had contracted a biotech company in South Korea to produce a human clone. In July 2002, the Raelians claimed that a young child, who they called Eve, was the first ever human clone. Now, again, there is zero evidence that Eve is a clone. They were never genetically tested, and the whole thing was just a tabloid story that got blown way out of proportion. All of this is to say that the scientific press in the US and UK were extremely careful about the reporting on the cloning issue, especially cloning stories coming out of South Korea. Although a certain amount of skepticism was warranted, reporting on Hwang's work still felt a little patronizing. Like they had to make clear he was one of the good ones. This didn't seem to faze the South Korean media though, who were simply ecstatic that a Korean was making headlines in the West. They took every opportunity to present Hwang's achievements as national achievements. Quote, Korean stem cell specialists were identified as heroes in their own time. No matter how the stem cell issue unfolds, wrote one commentator, they are on the cutting edge and medical experts from around the world will now be coming to Seoul to sit at their feet. A great Korean achievement. Koreans opened the way in organ cloning. Several headlines drew attention to one very particular goal. Hwang had a chance to bring a Nobel Prize to the nation. The Nobel Prize is a particular holy grail in Korean science. No Korean has ever won the prize for any of the science categories, which is in contrast to South Korea's perceived rivals of Japan and China, which at the time had 9 and 6 winners respectively. In particular, 2 Japanese scientists had won in 2002, which directly led to several committees being assembled in South Korea to figure out what they could do to secure their country a win. And the conclusion was obvious. Hwang was by far their best shot. 6th grade textbooks talked about him as a challenger for the prize. I cannot stress enough just how famous Hwang was. He used to brag to friends that he was so recognizable, he'd be swarmed in restaurants and compared himself to Korean Elvis. There was a genuine cult of personality developing around him. A fixation on one particular scientist, the likes of which hadn't been seen since the tragic story of Benjamin Lee. Benjamin Lee was a Korean-born physicist who had become the head of the theoretical physics department at a lab over in the United States. Bit of a big deal, place called Fermilab. Because of his prominence in the West, Lee was heralded throughout the 60s and 70s in Korea as a favorite to be the first ever Korean Nobel laureate. Tragically, however, Lee died in a car accident at the age of just 42. What followed was a widespread myth that eventually evolved into a full conspiracy theory among the Korean public. Because of tensions between the US and South Korea during the late 70s, it was rumored that Lee was bumped off by the CIA for his contribution to the South Korean nuclear program. The Korean media reported on this conspiracy for years, leading to several books and even a movie. In reality, Lee's work was completely unrelated to nuclear weapons, and Lee had openly criticized the South Korean government in the past. But that's the power of a symbol. Lee is beloved by South Koreans not because of his work, but what he represented. The potential to break through in the West. Hwang was fully aware of what was happening to him, and he played into it. Once when he was criticized for his aggressive PR, he simply replied by saying that he was, quote, an evangelist for biotechnology. He knew that adopting a nationalistic rhetoric for his work would only further elevate him, advancing his career. So when he made a cloning breakthrough, he made sure it was specifically a Korean cloning breakthrough. Remember that after his breakthrough cloning a cow, his immediate follow-up was to clone a breed of cow native to Korea and award the naming rights to the president. Next, Hwang said that he was engineering a type of cow that was resistant to mad cow disease, which was quite prevalent at the time. His goal, he said, was to help protect the Korean economy by making Korean beef more resilient. He then promised to, quote, spread the Korean people's spirit by cloning the Mount Paektu tiger. Siberian tigers and Korean wolves once used to inhabit the Korean peninsula, but were hunted to near extinction during the Japanese colonial occupation. Although he never managed to successfully clone these tigers, wolves, or cows, it didn't really matter. He got his message across. And these appeals to Korean nationalism, I would argue, were his more subtle attempts. Other appeals were a little bit more on the nose. During this period, concerns around mad cow disease prevented the import of livestock from the US. Hwang, however, would brag to the media that he had somehow snuck in pig embryos from the US and cloned them into perfect aseptic pigs. He then directly compared himself to Mun Ik-jom, a well-known 14th century smuggler who brought cottonseed from China. Now bear with me here, because this next one is going to sound very silly, but his most infamous piece of rhetoric was something called the chopstick theory. Hwang's cloning technique differed compared to other labs. When extracting the nucleus from the original egg cell, previous techniques used a needle to suck out the nucleus. Hwang's team argued that something about this subtly damaged the egg cell, preventing the cloning process from working. Instead, they began by making a small hole and delicately removing the nucleus by squeezing it with 2 metal rods. It was this change in technique that made all the difference. Hwang often compared the steel rods to the type of metal chopsticks used by Koreans for meals. To quote the man himself, "Who else in the world but Koreans can pick up beans with metal chopsticks?" When I show American researchers videos of my graduate students working with the micromanipulators, they are absolutely flabbergasted at their skill. The chopstick theory was an easily digestible soundbite that implied an inherent cultural advantage that Koreans possessed in the stem cell race. It played into international perceptions of East Asia as a place where labor involves delicate tasks like garment sewing. Even among other East Asian countries, the chopstick theory set Korea apart from its neighbors. See, the squeezing method used steel rods, unlike countries like Japan, which typically use wooden chopsticks. And finally, take this quote from an executive at Samsung. While leaving the task of basic science to the West and partly to Japan, we incessantly repeat the arduous work of processing and eventually discover better solutions. That's how we make money. It was this labor-intensive churn and the particular work ethic and culture of Koreans that had allowed them to become the world leader in stem cells. Western collaborators were more than happy to buy into this narrative. Quote, Korean scientists work much harder than Americans. Hwang's lab cloned and transferred more than 1,000 embryos into 123 dogs to make a single cloned puppy. That tells you how single-minded they are. If it was me, I would have given up after the 10th transfer. The atmosphere in Hwang's lab is very intense. Very, very intense. They're not just after the headlines. They just love what they do. Hwang was cognizant of the fact that he had two distinct audiences to appeal to, Koreans and the rest of the world, and so he shaped his messaging accordingly. To Korean media, he would assert that stem cell technology, quote, belongs to Korea. But when speaking to a UK reporter, he stated that, quote, I do not belong only to Korea. I have to do my work for all humankind. Those two statements read back to back are almost comical in how much they conflict. But Hwang didn't really see that as a contradiction. The duology of his attitude can be summed up by his paraphrasing of a famous Louis Pasteur quote. There is no national border in science, but a scientist has a fatherland. He was quite clear in his goals. I want to put the Made in Korea mark on stem cells. He proudly asserted to newspapers that his research would be able to feed Koreans in 10 or 15 years like Samsung did in the 2000s. He wanted to win Korea's first Nobel Prize at any cost. All of this is well and good, but there's still one question that was bugging me. How had a veterinarian with no prior international publications landed himself in Science, the world's premier journal? Well, he had found himself a partner, an American partner, and luckily, that American needed him just as much as he needed them. Getting into prestigious journals, although we like to pretend otherwise, is not entirely about the quality of the research. It plays a big part, of course, but there are thousands of quality papers that don't make it into a journal like Science, which again, only has an acceptance rate of around 7%. As a journal, Science is still a business, and it's in their interest to publish only the flashiest, most groundbreaking papers, and Hwang's stem cell bombshell certainly fits that bill. But there's another element at play here, a reason that a first-time author like Hwang could get into Science. It's because he had an even bigger name to vouch for him. Meet Gerald Shatton. At just 3 years older than Hwang, Jerry here was a big name in cloning, primate cloning specifically. Primates are of particular interest because they're the closest genetic relatives to humans. There's an extraordinary amount you can learn about human genetics and the treatment of diseases from studying other primates. He first made headlines in 1999 for producing identical twin monkeys using embryo splitting. In 2001, he led a team that produced the world's first ever genetically modified monkey, having spliced in a gene from a jellyfish. Later, he became the lead cloning researcher at the Pittsburgh Medical School, and he had made it his goal to produce the first ever non-human primate clone. But his work had hit a bit of a wall. His team had implanted 33 macaw embryos into surrogate mothers, and not a single one managed to produce a healthy clone. In 2003, he published a paper that suggested that cloning monkeys, apes, and humans may actually be impossible. During SCNT, when you remove the nucleus of a primate's egg cell, he posited that it unintentionally strips away two proteins that are essential to proper embryo development. Shatton seemed ready to admit that his years-long goal was out of his grasp. But later that year, out of the blue, he had been contacted by a Korean researcher who had supposedly done the exact thing he needed. Hwang was asking Shatton for advice to help with navigating the complicated review process at Science. According to Hwang, his team had submitted the paper to Science's rival, Nature, but had been rejected in May. He had followed up by submitting it to Science in June, but it had been stuck in an endless cycle of revisions for nearly 6 months. Hwang reportedly offered Shatton an authorship spot on the 2004 paper, but he declined. Instead, and I quote, Shatton volunteered to help steer the manuscript through acceptance by providing some editorial input into a revision which had already been written. He even called the editor at Science during the review process and, quote, lobbied hard for publication of this paper in Science without any direct knowledge of the veracity of the data. Shatton's vouching did the trick. Within a month of him getting involved, it was accepted. The paper had been sitting in the review process for over 6 months before he intervened. The thing you're probably wondering here is, what exactly is in this for Jerry? Why go out on a limb and tie his reputation to an up-and-coming Korean scientist that he's never worked with before? Well, it all stems back to Junior here.
Tonight I ask you to pass legislation to prohibit the most egregious abuses of medical research, human cloning in all its forms, creating or implanting embryos for experiments, creating human-animal hybrids, and buying, selling, or patenting human embryos. Human life is a gift from our Creator, and that gift should never be discarded, devalued, or put up for sale.
Jerry was doing his best to navigate a claustrophobic research environment under the Bush admin. In fall 2000, right before the mess that would be that year's presidential election, the National Institute of Health announced that they were accepting funding applications for embryonic stem cell research. When Bush finally took office just months later, he personally intervened and 180'd this decision. Make no mistake about this, Bush had made a tactical decision to couple the stem cell debate into a larger culture war, as a significant portion of his voter base consisted of evangelical Christians who were deeply opposed to any research involving embryos. This ideological lens was easy to spot when he heard the experiences some researchers had with the council. I presented our results in a very objective fashion to the politically appointed Presidential Bioethics Council, and at the end, I realized they didn't care about the science or the medicine. They wanted to know my personal religious beliefs. By 2004, though, the public conversation was evolving. Public polling was showing that the average American was becoming more open to the idea of cloning embryos year after year. Editor-in-Chief of Science, Donald Kennedy, weighed in. US politicians can't make the rules for everyone, and they don't have a special claim to the ethical high ground. If you couldn't gather Kennedy's opinion from his writing, the editorial was paired with a doodle of a nervous-looking scientist being monitored closely by a stern Uncle Sam. This cartoon was mocking the quote-unquote compromise solution that the White House had offered. The compromise was as follows. The funding ban only applied to federal funding. It still allowed for private and state funding, and researchers could still use the existing 71 stem cell lines that had been developed before the funding ban had taken effect. These 71 cell lines had been made using embryos, and the idea was, well, you might as well use them so they don't go to waste. Although these conditions sound reasonable on the surface, in practice they were still incredibly restrictive. Say for example that a university has a lab that is funded by an NIH grant. Now, even if the NIH funding isn't going directly to the stem cell research, it is going to the general overhead of running the lab where the research is taking place. That could get your university audited and you could lose your grant. What you had happening were universities building so-called safe harbor labs on their campuses, labs with completely different sets of accounting books just so they wouldn't get slapped with a lawsuit. Bureaucratic hoops to jump through that cost valuable time and lots of money. And as for the 71 cell lines, they had their own problems. Besides the fact that they were older and cultured using less refined techniques, the sample set wasn't genetically diverse. Only about 21 of them were considered useful. Certain diseases like Parkinson's couldn't be studied, and the cell lines weren't representative of a lot of ethnicities. Bush's compromise was the absolute bare minimum. The fears of researchers you heard earlier had come to pass. A brain drain that saw talented American researchers leaving for countries that would let them do the research they wanted. South Korea, of course, being one of the most prominent. So back to Jerry. He had fared better than most under the ban, since he primarily worked with primates, but the opportunity Hwang was presenting him with was too tantalizing to ignore. He could partner with an international lab, ensuring that all of the human embryos were only handled on foreign soil, completely bypassing the federal funding ban. Hwang and Shatton became extremely close collaborators. Quote, they met almost monthly, at one meeting or another, and they exchanged emails or phone calls almost daily to discuss scientific matters related to cloning. Shatton once called Hwang his metaphorical brother. It was a true symbiotic relationship. Shatton was able to research stem cells without Uncle Sam looking over his shoulder, and Hwang was able to borrow credibility from a well-respected American expert. Several members of Hwang's lab would visit and later join Shatton's team in Pittsburgh, including Pak Il-sun. She was highly valued by Shatton because she was well trained in the chopstick technique pioneered in the 2004 paper. With the help of her and others, Shatton's group adapted the technique to achieve the first ever cloned monkey embryos, 135 of them. None of them managed to develop into full clones, but the goal that Shatton had almost given up on was now back in the realm of possibility. As a thank you, Hwang was included as an author on the paper, although it's unclear exactly what his involvement was. Shatton had quickly become Hwang and Korea's #1 cheerleader. Quote, "We could have been struggling for decades. Now, our work is taking off fabulously. I think the whole world owes the Republic of Korea a debt of gratitude. I'm so proud of Korea and Korean people. I'm also so proud of the special scientist Korea has." By May 2005, their next collaboration was made public. Hwang's second ever paper with Shatton as co-author. The division of labor was clear. An actual line in the paper was, all experiments were performed in Korea by Korean scientists, and all results were obtained in Korea using Korean equipment and Korean sponsorship. Jerry apparently handled most of the writing. Once again, it was published in Science, and the larger community was dumbstruck yet again. There were two major improvements on display here. In the previous paper, they just kept trying over and over until they got a single stem cell. But for stem cells to be useful, you need them to be tailored to specific patients with incurable diseases. For this paper, they had stem cell lines from 9 patients with a variety of disorders, including spinal cord injuries, diabetes, and immune system disorders. The second achievement was a dramatic increase in the success rate of the cloning. The first paper needed 242 eggs to produce just one stem cell line, a success rate of 0.4%. This new paper used just 185 eggs and made 11 stem cell lines, a success rate of 5.9%. That's an improvement of 14 times. If the 2004 paper was like landing on the moon, this was like landing on Mars. The risk those egg donors were taking was paying off in a major way. This suggested that the technology was leagues ahead of where many thought it was. It was no longer in the realm of just R&D. There was already talk of clinical trials on patients. In one interview with the BBC, Shatton didn't hold back. The results were more significant than the discovery of vaccines and antibiotics and would rival the Industrial Revolution. The South Korean government responded with even more enthusiasm. They named Hwang the country's first ever Supreme Scientist, a title which came with a nice little cash prize of $15 million. All in all, he managed to swing almost $27 million in government support following his 2005 paper. A national campaign marketed him as the pride of Korea. The headlines continued to pour in. Korea falls in love with Hwang Woo-suk. One headline referred to Hwang as having God's hands. And speaking of God, Hwang had even been sharing his political opinions, voicing some sharp criticism of George W. Bush and his funding ban. Quote, "With all the great scientists and the great potential of the United States, if, because of some policy hurdle, the researchers cannot realize their dreams, I believe, as a fellow scientist, this would be very regrettable." Although he acknowledged that Bush had his own spiritual beliefs, he countered that by saying, "Let's say we have a microproduct just 100 micrometers long, made through nuclear transfer technology. And let's say we have a human being who has lived in pain all his life. To argue that there is a balance in considering the humanity of the two would not be ethical." If Hwang felt this comfortable weighing in on US politics, he certainly had no qualms about his own country's politics. You see, he had spent years making connections across all sectors of Korean society: the media, fellow scientists, and, of course, politicians. What people didn't see at the time was the stuff going on behind closed doors. Hwang had slowly but surely stacked the deck in his favor to the point that he was able to influence government policy. And he had built himself a loophole. Friends in high places. You gotta have them. And everyone wanted to be friends with Hwang. He was well known for inviting journalists out to his lab for personal tours, but at one point he hired Yoon Tae-il to be his PR man. Yoon Tae-il was an ex-journalist and high-ranking director who had worked for YTN, the first ever 24-hour cable news network in South Korea. Over the years, Yoon Tae-il would use his connections to help shape glowing headlines, and as we'll see later, suppress negative ones. Next, the names that make it onto a scientific paper are as much about actual contributions as it is about networking. The author list on even some very good papers are often padded with names who never even set foot in a relevant lab. Of course, you know Gerald Shatton already, but Hwang had built up a roster of prominent Korean scientists to hype up his work to the media. Take An Curie, She was the first ever female doctor at SNU's Department of Internal Medicine. She had goals of using cloning to help solve Korea's organ donation crisis. She often acted as a deputy for Hwang and would speak to the media if Hwang himself wasn't available. Then you have Moon Shin-young. On the 2004 paper, he was awarded the coveted final author spot. Moon was a well-respected professor and director of the government-funded Cell Application Project. When the matter was later investigated, it was found that Moon's involvement in the paper was slim. He provided limited technical assistance and some editorial advice. A prestigious rubber stamp, nothing more. Another key name on the 2004 paper was bioethicist Park Ki-young. It's unclear what work she did for the paper. At the time, it was pointed out that her background was in plant molecular biology, not human biology as you would expect for a paper on human stem cells. It's been alleged that she was added to the paper to give it political weight, as at the time she was the manager of the Presidential Advisory Council on Science and Technology. Park's political connections led to the formation of an under-the-table policy group that included the National Policy Secretary and the Information and Communications Minister. The four of them being so close was picked up on by the public, and they came up with a nickname that combined their four surnames, which, when translated, roughly means golden bats. Gotta say, if your weird little under-the-table political influence group has a nickname comparing you to bats, you're probably doing something sketchy. And yes, sketchy things were happening. The IT minister had awarded Hwang a research grant worth $4.3 million from the, get this, Advanced Backbone IT Technology Development Fund, even though Hwang's research clearly had nothing to do with IT. But because Hwang was the nation's supreme scientist, no one batted an eye. And of course, Hwang's personal network eventually expanded to include the biggest prize of all. Park later received a promotion and was appointed the personal science and technology advisor to the president. Yes, in June that year, Hwang was awarded a medal directly from the president himself. And it was this network of influence that allowed Hwang to shape a national law, South Korea's Bioethics and Biosafety Act. As we discussed earlier, South Korea had its fair share of bad PR due to cowboy cloners and UFO cults. This was because between 1997 and 2003, South Korea technically didn't have any regulation that prevented human cloning. There was a massive legal gray area that was ripe for abuse. It wasn't for a lack of trying, though. Several bills were put forward by individual politicians in this period, but none made it past a successful vote due to disagreements between the ruling and opposition parties. The government, first under Kim Dae-chung and later under Roy Moo-hyun, wanted to thread the needle in such a way that cloning would be restricted, but with enough exceptions that it could still boost the recovering economy. The only way a bioethics bill would pass is if it was drafted by the sitting government. So in early 2000, the Kim administration tasked its Ministry of Science and Technology to come up with proposals. By definition, the ministry is part of the existing government, thus naturally it will have a slight bias in favor of their priorities. However, in the year 2000, the ministry created KBOC, an independent bioethics advisory commission consisting of half scientists and half non-scientists. In a move that caught the ministry by surprise, KBOC came back saying that both reproductive and therapeutic cloning should be banned, with only a small exception for excess embryos made through IVF. This was clearly not the result that the ministry was expecting, and rather than draft a law that they knew would be unpopular with both conservative and liberal politicians, they decided to just not do their job and never ended up submitting a bioethics law to be voted on. The government was then like, okay, I guess we'll get someone else to do it, and gave the job to a different ministry, the Ministry of Health and Welfare. But this also backfired on them because the Ministry of Health and Welfare just ended up agreeing with KBOC. It was a mess. You had 3 separate bodies weighing in, each with their own take on the law. The government had found itself in a no-win situation. Hwang had been saying in interviews that if the law didn't go his way, he'd simply pack up his team and just go work in a different country. And so, the government stalled and stalled for nearly 3 years until the National Assembly pushed through a version of the bill a couple days before New Year's Day 2004, although the bill would only take effect a year later on January 1st, 2005. Because of this, Hwang's 2004 paper was technically not legally bound to the act. It could skirt through within the legal buffer period. It has been alleged that the 1-year buffer period was added to the act at the suggestion of Park Ki-young, as she would have had a large amount of influence over any science policy. Remember that she was added as a co-author to the 2004 paper at the last minute, and she had worked on 2 other projects for Hwang, and for those, she was paid $250,000. At a glance, the bill seems like it addresses most of the concerns of the bioethicists. Human reproductive cloning is outright banned. Human-animal hybrid research is banned. And it outlaws the selling and trading of human sperm and eggs. And any violations could result in criminal prosecution. And most importantly, cloning of human embryos via SCNT was now banned. But there was a catch. If you had previously been conducting embryonic cloning research, you could continue, so long as you met 2 conditions. Condition 1, you must have been working on the research for more than 3 years. Condition 2, you must have published relevant research papers more than once in relevant journals. At the time the law would go into effect, this exception would only apply to 1 lab in the entire country, Professor Hwang's team at SNU. The South Korean government had passed a law with a loophole specifically designed for 1 man. A few other labs that had the necessary scientific expertise were arbitrarily blocked because of this clause. By bending the rules for their pet scientist, the government had willfully laid the groundwork for what would soon become an international scandal. As spring 2005 turned into summer, Hwang announced yet another breakthrough, this time returning to his animal roots. His lab was the first in the world to clone a dog, which prior to this was considered one of the most difficult mammals to clone. Specifically, it was an Afghan hound named Snuppy. The global media attention on Hwang was so powerful that in October 2005, the South Korean government launched the World Stem Cell Hub, a research network to be directed by Hwang where international collaborators could partner with South Korea, funded through nearly $25 million in government grants. Countries that weren't legally able to study stem cells at home could still contribute by having Hwang's team carry out the work in South Korea. Alongside Hwang, 2 members on its board of directors were Gerald Shatton and Ian Wilmot, one of Dolly's creators. Truly, Hwang was now in the big leagues. South Korea was no longer an upstart nation playing catch-up. It was a veritable biotech superpower. But 1 month later, Gerald Shatton would pull out of the World Stem Cell Hub. And 1 month after that, he'll retract his name from the 2005 paper. Soon, South Korea would be racked with wide-scale protests, the resignation of high-ranking government officials, and Hwang Woo-suk, the cloning king of Korea, would be under criminal investigation. There was a darker side to Hwang's lab, and it was all about to come out. All because of one seemingly innocent question. Where did you get those eggs? Hey, are you dying to watch part 2, like right now? Well, you can watch it early right now on Nebula. Nebula is the creator-owned streaming service I'm a part of alongside some of the best educational creators on the internet. There you can watch my videos and many more completely ad-free. Not only do I post my videos 2 weeks early there, but you can also find dozens of Nebula originals not found anywhere else. If you're looking for a deeper dive on the Korean Peninsula, I'd highly recommend the Modern Conflicts series by Real Life Lore. Or if you want more on the medical side, I'd recommend Common Diseases Science Still Can't Cure with Joe Scott. Maybe you're looking for advice on making your own videos. For that, there's Nebula Classes, courses by some of the best creators out there, from the basics of video editing to elevating your videos with great audio. And here's one more major reason to support me on Nebula. As of this video, I am officially a full-time YouTuber. That's right, making these videos is my full-time job now, and signing up to Nebula using my link is one of the best ways to help me out. With it, you get a 40% discount on an annual subscription and access to Nebula Classes, all for just $250 a month. And a significant chunk of that money goes directly to me for as long as you're subscribed, which will help me make bigger and better brockumentaries. As always, thanks for watching.